So today, it is my great pleasure to introduce Cassie Mitchell. She's a new faculty member in biomedical engineering, but she is not a new face here. I had the pleasure of being on her qualifying exam committee as a graduate student in biomedical engineering.
You're showing our age, Lena.
Yeah, showing my age. She was on our, a fellow on our NSF IGERT program in hybrid neural microsystems, and then she got her own individual NSF fellowship. So for all of you writing your applications right now, this might be good. And she then, I think, got a paper out of her project, her first semester in graduate school. She's been breaking all kinds of barriers since she's been here. Stayed, got a K award, R awards on her own before now being on the tenure track as an assistant professor here. And over those years, she's taken on, as she has in her whole life, a lot of sports endeavors. I've seen her go through rugby and cycling and discus and swimming. And she has, many of you may know, Olympic medals, Paralympic medals from the last cycle, right?
2016?
2016,
yeah.
Bronze and silver, and many world championships in a large number of sports. So beyond that, her work is here that you're going to hear about, I think, is really original, and you're not going to see an approach like this. She's really forging this field. So anyway, I hope you take the opportunity to meet with her afterwards, and you're welcome. And new faculty, but really somebody who exemplifies, our program and someone we're really proud of at Georgia Tech.
Thank you, Lena, for that warm introduction. It's an honor to speak to you all here today. One of the problems about being at a place for a while is, ironically, you kind of start to blend in, and then you don't have the opportunity to talk to the up-and-coming students. So it's amazing to have such a large audience here today to hear about the work that we're doing in the laboratory for Pathology Dynamics. Okay, so I'll hop right to it. The title of our talk today is After the Ice Bucket, Thawing Amyotrophic Lateral Sclerosis with Predictive Medicine. Because we have students in the room, I think it's prudent to discuss a little bit about things that aren't research. Sometimes when you're in the grind with research, it's easy to get carried away and kind of miss out on the other things that happen in life. Lena has shared a little bit with you about me, but I think as an engineer, we often hear about first principles in science, first principles in engineering, how we get our equations, but we also have our first principles as people. And as a researcher, those first principles guide you not only in your science, but in your life. And so I think regardless of what your first principles are, it's important to have some, because if you don't, then you will be molded into somebody that's not yourself. Anybody that's been in my office will know that I have a sign that says never, never, never give up. That's probably my #1 guiding principle, that it goes around everything else. That's a good one for researchers because you get rejected a lot. I also am a mentor at several different Atlanta hospitals. I do patient counseling, mostly for neurological disease, but also a wide variety of other diseases. My faith and my family is important, and as my students know, they are also very important to me, which is why I have a large lab. All right, my career path, just the elephant in the room. Yes, I am paralyzed. I'm in a wheelchair. I'm a C5-6 level quadriplegic, but I wasn't always in this chair. And maybe one day I won't be, because amazing researchers like yourself will have me get right on up and walk right out of this room. That would be awesome. But I was paralyzed at 18. Before that, my first job was as a racehorse jockey. The farm next to us had horses, thoroughbred horses, and I raced, and not like for myself, but for the farm next door. And eventually, I got this horse here that was off the racetrack that had been injured, and I rehabbed her, got her really cheap because she'd been injured, and we went on to win. That was my first world championship, actually, was in rodeo in a sport called pole bending. You guys can look that up afterwards. At 18, I was paralyzed right after that world championship. If I would have been paralyzed because I fell off my horse, that would have been somehow more cool than the way I was paralyzed, which is with neuromyelitis optica. Any of you guys that are going into neuroimmunology, you can maybe do some research into that. But it affects the cervical spine and the optic nerves, causes vision changes as well as paralysis similar to a spinal cord injury. I went on to Oklahoma State and got my bachelor's degree in chemical engineering, went to ExxonMobil. I worked in big oil, made really good money, made more money than I make now. But money isn't everything in life, and so I decided to become a pauper and go to graduate school. You guys probably know that feeling. Best decision I made in my life. I've really enjoyed the work I've done here and with my students, and so that leads us into the next aspect of my life that Lena mentioned, which is my athletic career. This is kind of the perpetuation of never, never, never give up, all right? So as my disability changed and I became more disabled as the disease progressed, I switched to many different sports, but whatever sport I went to, I wanted to be the best and to do the best. And that's kind of like in research, right? Sometimes the questions you go after, they don't work out, and you end up with a different question than what you thought, but you keep forging ahead because whatever insight you get is helpful to the patients that we are trying to help. All right, so moving on to today's topic, amyotrophic lateral sclerosis. So back in 2014, there was this thing called an ice bucket challenge, and everybody all over the world was dumping buckets of ice on their head. A lot of people didn't really know why they were doing this. It was just like the cool thing to do, and it went viral. But it was for a disease called Lou Gehrig's disease. Here in the US, we call it Lou Gehrig's disease. This is a picture of President Peterson here at Georgia Tech, and my students were very happy to dunk him with a lot of ice water, being our representatives of ALS research on the campus. But anyway, back to Lou Gehrig's disease. Lou Gehrig's disease was named after a baseball player here in the US, very famous back in the 30s. Lou Gehrig was diagnosed with ALS at age 36. An interesting fact is he was diagnosed by Dr. Charles Mayo, which many of you have heard of the Mayo Clinic up in Minnesota. His records still reside at the Mayo Clinic. And Mr. Lou Gehrig, he died within about 2 years of his diagnosis, but he was an amazing advocate for the disease. And to this day, his family still are big supporters of ALS research. Also, he still holds many records as a baseball player, even playing back in that day. All right, a few facts about ALS. If you go to the ALS Association, that's the same association that sponsored the Ice Bucket Challenge, you'll see that there's about 5,000 new cases of ALS every year. It's, relatively speaking, considered to be a rare disease, but it's one of the more prevalent motor neuron diseases, meaning stuff that actually affects the motor nerves, which connect from the brain to the muscles, all right? Unfortunately, the average time of death is between about 2 and 5 years. There's only 2 FDA therapies that are currently approved. One extends life from about 1 to 3 months, and the other 2 to 4 months, all right? So it's pretty bleak right now. Only about 10% of the cases are familial, meaning that there's a known family connection. And as you guys know, there's a difference between familial and genetic, right? Familial means you know that you inherit it. Genetic, it could be like sporadic mutation, and not necessarily just an inherited mutation, all right? But only 10% of the cases are familial. And, you know, because it's a rare disease, it does not get as much research funding as something like Alzheimer's. The symptoms of ALS are varied. You have 2 main types of onset. There's a limb onset, which is closer to the neurodegenerative diseases we're used to, where you have weakness in the extremities. You may have fasciculation similar to Parkinson's disease. The other type is the bulbar onset, which affects the pseudobulbar area of the brain, and it causes problems with swallowing, clinically called dysphagia. It also causes problems with speech, particularly dysarthria, which is how they formulate the words, usually due to the facial paralysis and caused by the facial nerves that are affected in that onset type. And then about 2% of the patients get what we call a respiratory onset, which means that their intercostal muscles that help with breathing are affected first. Ultimately, patients usually die due to respiratory distress, the inability to breathe, or to secondary complications from respiratory problems like pneumonia. All right? ALS is very complex. Whether you have an inherited version of the disease or whether you have a sporadic version of the disease, it's not just one gene that dictates what happens, all right? It's lots of genes and lots of cellular pathways. And it doesn't just affect the neurons, all right? It can affect also the supporting glia, the muscle tissue itself. And in fact, there are many different theories that say that ALS can be started either from the spinal neurons or from the neuromuscular junction or the muscle. So you have like all these different touch points to where one part of the physiology connects to the other. Energetics pathways are definitely impaired. A lot of patients will lose a lot of weight. Some of that's from the dysphagia, but it also seems to induce a hypermetabolic state. Early in ALS, you'll also have some excitability issues. They've noted like excitotoxicity from glutamate and other neurotransmitters. So it's just a very complex disease, and that makes it very difficult to study because you can't look at all these things at once in a single experiment. All right, the number one experimental model for ALS is the SOD1 G93A, all right, and I'll try to spit this out, but I'm not an experimentalist, so you'll have to forgive me. But superoxide dismutase 1 glycine 93 to alanine, that's what that stands for. And what that does, it's one of the superoxide dismutases that helps in free radical destruction. So when you undergo respiration in the mitochondria, free radicals are produced. You can also get oxidative stress from other sources, like the sun, which is why we wear sunscreen. But whether it's internal oxidative stress or external to the cell, free radicals have to be removed, and SODs help to do that. And in the SOD1 G93A model, the SOD1 G93A protein is not properly folded. So what happens is, instead of being this nice tight little wad that it's supposed to be in its active form, it is misfolded, so it's like a big bunch of junk, right? And so then it kind of just junks up the cell. And when that starts to happen, not only do the free radicals not get destroyed, but transport is impaired. Things don't get where they need to go. The high copy mouse, which is the most common one in the literature, its onset is about 85 days, and the average time of death is about 150 days. Mostly we use functional metrics to characterize the disease, like rotor-rod, grip strength, and body weight. And then, of course, you have the actual metrics used to quantify the cellular pathophysiology itself. All right? So moving on to what my lab does to try and help ALS. My lab, I like to think of us as doing predictive medicine. So whether it's in ALS or another neuropathology or even something that's not neuropathology, all right, we have three main goals. The first goal is to identify multiscalar disease etiology, all right? So to figure out what's exactly going on, especially in the case of multifactorial disease, right? Where there's multiple causes that could be triggering this disease. The second thing we want to do is to be able to predict new therapeutic strategies, right? We all know that FDA things take way too long, like 7 to 14 years, sometimes even more. So if we can try to predict up front what looks like it's going to be successful, then that helps get therapies to the patients faster. And then lastly, as a patient myself, not with ALS, but with another neurological disease, I'm all about wanting to optimize the quality of life for patients now. So maybe I can't cure a disease, all right? But I want to make them live as long as I can and make it as comfortable as I can for them for as long as possible. And sometimes I think as researchers, especially in neuroscience, we tend to forget about that. We tend to want to, have the magic bullet that cures the whole thing, when really there are incremental things that we can do that help patients today. So I try to have that be a third aspect of my lab that's important. All right? So being a lab that works with data, all things data, we have to have this balance between big data and smart data. I know there are lots of big data people in the room, and those big data people are bigger than I am, okay? So the data sets that I work with, we're not to petabytes or anything like that, but they are large data sets, all right? Eva, who's in the room, she far outweighs me on the size of our data. So that teeter-totter would go the other way, to call her out on that. So, but then I can also tease it. Maybe I'm more on the smart data side, right? And that sounds cool. Well, what is smart data? Smart data means that you're looking at the relationships within the data. So it's not just the number of measurements that are taken and their reproducibility, but rather the unique interactions and interrelationships. So it's always highly dimensional, all right? So instead of having 1 million observations that you have 10 metrics of, you have like, 1 million different metrics that you have 10 observations. So it kind of reverses the matrix, all right? It's a very dynamic, all right? Time is a very important part to what we do. And so these are all continuous models. All right, so that's very different than the traditional big data, which focuses on everybody's favorite in this room, which is electrophysiology, right? And then you also have genomics, imaging, and proteomics. But there are all types of data that move beyond these realms, and that's, my lab does work with these types of data, but we want to extend informatics to beyond those types of data, all right? To the standard, I measured glutamate in an SOD1 G93A mice, and it went up or down. And then I applied this treatment, and then it went up and down. There are lots of other types of measurements beyond these 4, all right? So we call this pathophysiology informatics, all right, which is the application and integration of in vitro, in vivo, and in silico data to assess complex disease etiology, progression, and treatment, all right? So that goes along with the 3 goals of the lab, which was to identify what's going on, to help treat it, and to make things better, quality of life. So it requires big and smart data. But the 3 things that are unique about the type of data that we work for, other than what I've already told you, is that the data sets are scattered. So there isn't one prospective data set where one lab has measured all the things about ALS, and therefore it's nice and controlled and very clean. It's not like that. Most of our data sets are scattered across numerous individual journal articles, where one scientist measured one thing, another scientist measured another thing. Because of the different types of data, it requires a lot of diverse analytical protocols. You can't always analyze every piece of data the same way. All right? So I call this the knitting of the quilt, all right? So when someone makes a quilt, I know that's kind of like old school. Probably nobody in here knits or makes quilts, but just go along with it, all right? Again, showing my age. I'm really not that old, but anyway, let's just go with the quilt, all right? So your grandma made a quilt, all right? And she started by, stitching together these little patches. And these patches each represent, say, a scientist's point of view, one journal article. So when you look within that one patch, you do learn about what's going on in ALS, but it's not the same as when you stitch everybody's patches together, and then you can start to see a pattern, right? So that's what we want to do, is take all the journal articles and stitch them together so that we can get a comprehensive view of what ALS is doing. All right? We call this viewpoint aggregation. So you have your quilt, right, which is in the green circle there. If you haven't noticed, green is my favorite color, all right? All right, so we have the green circle that has the quilt. And then what we do is we swap out pieces of information. So as we get more intelligence about that aspect of the quilt, we can put in an actual mechanism, like a mechanistic model, like your Hodgkin-Huxley model, right? So you can insert more knowledge as you get it. You can also insert clinical treatment predictions from clinical data. You have your experimental observations, and then you also have your conceptual insights. So conceptual insights are things that we understand, but we don't necessarily know what the mechanisms are. All right? And the idea of being able to stitch this quilt together comes from complex systems theory. All right? This is what my PhD thesis was about, is being able to use complex systems theory in order to characterize multifactorial systems. All right? So there was this awesome paper that was published by Dr. Setti and Doyle. They were actually at Emory University. They may still be at Emory University. Lena, do you know if they're still there?
No, Caltech and
Okay, Caltech and Craft, for those of you who didn't hear that, all right? So I don't know where the Craft University is, but I'll leave it at that. It's an awesome paper, and it really inspired my PhD research. And you guys may have a paper like that too, where it's like, wow, I read this paper and it was just so cool and it really got me going. Well, this was the paper for me. And basically, they had this theory called the bow tie theory, where in a complex system, and they were looking at like cellular metabolics, all right, like in yeast cells and that types of thing. But anyway, you have multiple inputs that come into your system, all right, and then you have these local relationships, and those local relationships specify this really complex adaptive behavior called emergent properties, all right? And emergent properties, the most famous example that neuroscientists love is the action potential. No one calcium channel or sodium channel causes an action potential. They all work together in order to have this emergent property, which allows neurons to work. Well, within these local relationships, most of which can be experimentally measured, you have dimensional restriction, all right? So it's a pinch point, and because of this, it looks like a bow tie. And so that's how the bow tie theory was formed. All right, so this idea of using local relationships, which can be experimentally measured in order to dictate system behavior or predict it, that's what the lab is founded on. All right, so another concept that we like to use a lot is system stability. All right, so all know how hard it is in your neuron models to try and get something to be stable, to not oscillate, and then you have things like bistability and all this types of stuff. So you'll understand that stability is an important part of neurons, right? But it's also an important part of all of physiology, all right? When we learn that like in second grade when we talk about homeostasis. So one of the things the lab tries to do is use the fact that all physiology is like its own system, all right? And there's a rate, a gain in between each pathway in the system, and those gains work collectively to dictate how the system responds to perturbation. So you have a perturbation, and if you're not in a disease state, you can respond, but if you're in a disease state, you're not able to respond properly. So an instability is propagated, and as long as the instability is there, the disease continues to progress. All right? So here's another QT thing that will help you remember what our lab does. Access, aggregate, and analyze. That's the AAA approach, all right? So if your data is broken, who do you call? Call the Pathology Dynamics lab, all right? Not AAA. Car's broken, you can call AAA. If your data is broken, call us, and we will help you figure it out, all right? So I have to be careful what I say here because my first PhD students in the room, call Andrew out here, and he's going to feel kind of not loved right now because I'm going to talk about the undergraduates for a little bit. But I have moved beyond just undergraduates, so anybody who's interested in PhD research now or later or whatnot, you can spread the word. But we'll talk about the undergraduates for a little bit. I started with undergraduates largely because when I started my lab, that's what I had access to, all right? And I needed a lot of people to do what I wanted to do. It takes a lot of people to gather all this information from different journals and look at all these different aspects of the physiology. So I created my own little corporation, and it runs very smoothly. People are trained, and they have these apprenticeships, and people enter data live. We have our own quality control. We have our regulatory. It's like different departments within the lab, all right? Some of these people are interested in research. Some of them want to go to industry as project managers. In any case, we run little pilot projects about different aspects of the physiology, and that helps drive us like where we need to go next, right? So we take in data, we can prioritize like what we need more data on, what we can analyze now, and then we're building these comprehensive models. And then it all has a little management system wrapped around it, all right? Brag about my undergraduates a little bit. They've done really well. They've probably had more first author publications come from our lab than some labs have from PhD students. They work really hard, have great acceptance rates into medical school and into industry. And a lot of people have gone on. I think my first set of undergraduates graduate medical school this year or finish their internships and all that past medical school. So now, again, showing my age. But they've done really well, and I'm really proud. All right? And I know that my graduate students will do well too. So put that in there, all right? So next, you have your system in place to aggregate your data or to access your data, all right? And if you want to set up your own little undergraduate corporation, I'll be glad to tell you how to do that. But we use 3 types of techniques. First is the human workflow where you have lots of people, all right? And you have to like optimize the human aspects of the work. So that comes with training, that comes with quality control, all right? We also have partial automation, where you have different types of tools to help optimize the workflow of the human part. So for example, a field that's listed as, numeric only, or like if you don't fill out this field, you can't move to the next page of the database when you're entering data, all right? And then we also have full automation, which is what we're moving towards now, which uses like optical character recognition, natural language processing. Those are kind of the future. To be able to go to each of these journal articles, gather what you need, and put it into a database that you can then analyze, all right? So doing a completely manual approach, all right, you can start by getting your documents, downloading your PDFs, then you gather all your figure information, and then you get your data series, which data series just means like you have something plotted X, Y. I'm going to grab the trend line, and I'm going to pull off those data points, and then the response value is like one data point. Glutamate is this concentration at 30 days, right? And so depending on how much data was in an article, it takes about 100 to 200 minutes. With partial automation, it's closer to 100 minutes, all right? But full automation definitely needed to hurry this process along when you have thousands of articles, all right? So this is an example of one of the databases that we've developed for ALS, which you can access on our website. And so it basically uses the structure of the journal articles, because the experiments are so different between different journals. You have to use the structure of the actual article in order to have the commonalities between the data to make it work in the database, because everybody plots their graph in X, Y form, or they should, right? And so, so that works really well. All right? Normally, biocuration, that's what this whole process is called. When you are taking data from different sources and compiling, it's called biocuration. It's normally done by PhD students, but even with the undergraduates who've been properly trained, our accuracy is around 99%, and probably about 97.5% before quality control, and with quality control, it goes above 99%. So the system works really well. All right, another thing that we do is clinical record curation. All right, so all of you aspiring clinicians, whether it's clinical research or you want to be a physician, one of the big misnomers in medicine is that you have electronic medical records, but those aren't really electronic. They're just scanned forms that somebody has filled out and put in. So we're developing methods in order to curate ALS data. So you have, for example, the muscle scores, you have what medications they're taking, you have their forced vital capacity, different metrics that characterize clinical ALS. Right? The next step is to be able to aggregate your data. You have gone to all the disparate data sources, whether it's clinical or whether it's experimental, and you got into a database. Now what do I do? All right, now I got to aggregate apples and oranges, all right? So there are two main types of aggregation techniques. There's physiology-based, which is like I understand that glutamate is part of excitability, and therefore that's like a concept-driven. Or there's data-driven, where I use something like unsupervised learning or something along those lines in order to figure out what the data says apples to apples are, okay? And so the long-term goal is to get an ontological field map so that each colored box kind of represent things that are related, that they kind of go together. And then you have hierarchies of factors, right? So within apoptosis, maybe you have like caspase and BAX and BCL2, and anyway, so this picture depicts like the different journal articles, right? You have complex physiology, then you get a map of where the disease is. The bar graph represents kind of the distribution of data for each of the different ontologies. After you get your ontologies, the next step is to get your relationship map, all right? So I have all the boxes, little ones, big ones, and how they're related, but now I need like, what's the magnitude of the relationship between this box and that box, right? How many boxes should I even have, right? And so that's dictated by the amount of resolution you want. How many journal articles do we need to aggregate to get a statistical significance on a specific relationship? So then we start to figure out different schemes that work, different maps that work for showing what the disease looks like. Looks awful lot like what I saw when I was a chemical engineer at ExxonMobil, when you had your plant, right, and all the systems and the feedback. And that's kind of what we model it after. Once you aggregate your data, you can start to see trends. So before you even do anything fancy, you can already see in this picture that there's a difference between the blue and the green dots. And before this analysis was done and published, people in the field thought the green and the blue were the same. Those are actually two different types or strains of mice. People used to think that gender was more important than strain. And just by aggregating all the data from 703 articles, we were able to show that strain was actually more important than gender. And a very common metric in ALS, which is rotorod. So even beyond doing the fancy techniques that I'll talk about in a minute, you can still just aggregate data, do traditional analysis, and get very important conclusions. You can also do the same with patients, all right? So here we have the probability of survival of ALS patients over time. And you can predict survival using both quantitative and qualitative metrics, all right? But you have to determine how you're going to aggregate them. Right, so, right, moving to the analyze phase, this is kind of where all the excitement happens. There's a lot of work. So it's kind of like when you're cooking your pie, chopping the apples and all that stuff. That takes a lot longer than just putting it in the oven. Analysis is kind of like just putting it in the oven, all right? We got all of our data in the form we want. We got it all aggregated. Now we get to actually bake the pie. And this is the analysis. So the goal is to be able to kind of come up with a weather forecast of what looks the most promising in ALS or whatever disease that we happen to be working on. And you have forecasts for theoretical, experimental, and clinical. All right? We use traditional statistics. We use de novo systems modeling, mechanistic modeling, and also machine learning and optimization. And we also use them in combination. One of the most interesting things that we do in this lab is called dynamic meta-analysis. It's kind of what we're more known for. And this is where we take that relationship map I showed you and use the idea that this is a system that has to be stable, and we model it over time using both the wild type and the treated G93A mice data. And then we use the fact that it should be stable. If it's a wild type mouse, it should be stable. At ALS, we can look and see what kind of instabilities may be there. And when we did it with a 10-way ontological model, we could see that the oscillations actually occur well before symptom onset. If symptom onset's at 85 days, we actually have oscillations at 30 days. So you can come up with these new treatment paradigms where you can actually, instead of trying to fix glutamate or fix apoptosis or fix SOD1 G93A, what if we just try to re-stabilize the system after onset? Because when we get patients, we don't get to know they're going to have ALS up front, right? We have to fix them after they've had onset. So we tried and seeing like what relationships do we need to tweak in order to not like just to re-stabilize the system as is, irrespective of what caused it at the beginning. And this is a unique idea. You can use the eigenvalues just like you normally do in system stability to predict. And it turns out that there are a couple percent of the 100,000 treatments that we tried that actually are stabilizing. So then those treatments would be the ones that we would suggest that would need to be like experimentally verified and then go through the stages. But you can see that of 100,000, 2%, right? So this is why it's important to have informatics so that we can predict up front what looks to be most successful. I'll move through here a little more quickly. Most of you guys are familiar with experimental meta-analysis. That's something that we do a lot in our lab. If you ever need help with that, kind of how can I aggregate my data or other people's data to do simple trend lines, let me know. Something interesting on this slide is on that bottom, your right there, you can see that there's oscillations in the data even without putting on top like an actual model. It's just there already in the experimental data. So that's kind of interesting. We can also do something called multi-treatment meta-analysis where you aggregate all the treatments and see how they work over time, all right? And one of the interesting things about multi-treatment meta-analysis, obviously this is in mice, is that sometimes when you actually give a treatment is more important than what the treatment is or what category it came from, all right? So something like riluzole, which is supposed to be like good for ALS, it's sodium channel blocker. If you don't give it early in the disease course, it actually doesn't work. And in the mouse model, we show that if you actually give it later in the disease course, it can actually make the ALS worse. So it's not just what factor is being perturbed and what you're trying to fix, but when. because these relationships change over time because it's a complex system. So the relationship isn't the same throughout the disease course. All right? We also do epidemiological research to try and find commonalities within patients, like what experiences do patients have that might describe, why they were more likely to get ALS. So if you are diabetic, all right, or you actually have a lot less chance of getting ALS, so that's kind of interesting. If you have liver disease, you have a less chance of getting ALS. But if you have, certain other things, in general, ironically, the more healthy you are, the more likely you're going to get ALS, which is this weird oxymoron, and we call it the hypervigilant regulation hypothesis, which basically states that because the ALS system has such high feedback gains that they actually tend to overcorrect people who tend to get ALS. And if you tend to overcorrect, like any high feedback gain, that's good as long as the perturbation is small. But if the perturbation becomes large, it generates what? An instability, right? And so that's why people who tend to have less antecedent disease, we think tend to be more likely to get ALS. There's lots of overlap between ALS and other disease. So you compare different biomarkers, and we can look at that. There's huge overlap between ALS and AD, which means that using complex systems approach, we should be able to have shared therapeutic ideas. We've also done work with quality of life interventions like bi-level positive airway pressure. This is one I have in review that I'm really excited about. Bi-level positive airway pressure is basically this mask you wear, kind of like a CPAP that people wear for snoring. And what it does is it forces air into patients' lungs, without actually having to be like with an internal tube, all right? And basically because of the way the insurance right now, you can't get it unless your forced vital capacity is half that of a normal person. And if you do that, basically the survival is almost less than half of what it would be if we would allow patients to get in a lot earlier when it's like at 80% of predicted, which basically means that they need to be wearing BiPAP before they have like really substantial breathing symptoms. But when you do that, then you actually get more survival duration than what you do with the FDA-approved drug Riluzole, which is pretty cool because BiPAP doesn't have the adverse effects that Riluzole has. So that's an example of we're trying to optimize, and we actually find something that's better than another treatment that was trying to, quote, cure or, slow degeneration. All right? One of my favorite analyses for any type of data is cross-correlation analysis. You can make these nice dendrograms where you can see kind of what different metrics you have are related. It's another way to aggregate your data. It kind of helps you see visually not only what's correlated, but how can I aggregate different data sets, okay? And sometimes things don't aggregate the way we think. For example, the ALS-FRS score has both a breathing score and a limb score, and they combine it into one score. But when you combine it into one score, the reproducibility on it is actually less than if you left them separate. So one of the recommendations we're trying to make right now is that we don't combine it into one score, that we leave it as two. And then these type of techniques help enlighten us because sometimes A plus B just does not equal C. Principal component analysis, I'm sure a lot of you guys are familiar with this. It's great to see the variance in the data, kind of predict how many dimensions are in your data. Do I have enough measurements to really characterize the variance in my data set? Machine learning models, which I know a lot of you guys probably know more about than I do, but we do a little bit with that too, to try and come up with models of ALS survival. And reduced models so that you don't have this overfitting problem that often comes in with machine learning. And when we do that, we found that duration and age actually is the most predictive, more predictive than muscle scores, ALS-FRS, weight, height, and sex and onset type. And that goes totally against the kind of the clinical dogma, if you will, of what's important in ALS. So age and disease duration tend to predict survival the best. We can also classify patients. This is a new model that we have where you can kind of predict within 30, 60, 90, 1.5 to 3 years how well patients are doing, and it combines both qualitative and quantitative data to do this. All right, so I'm kind of heading towards the summary here. This viewpoint aggregation approach, where you take clinical, experimental, theoretical data and you put it together, it can result in these neat theories like hypervigilant regulation. And so those type of theories kind of let us use engineering systems in order to come up with better therapies. Instead of trying to fix the initial perturbation, I'm trying to restabilize the system. We definitely need better clinical metrics to reduce heterogeneity in the data. That's a big problem with ALS, just because there are so many different factors that go into it and also what causes it. And then we obviously need more optimized interventions. All right, getting ready to go for a landing here. So the future of predictive medicine. Definitely need automation, all right? Big data, automated biocuration. And any of you guys who have to sit and annotate data, who do like machine learning, you understand this. We need some automation to help us. And I think natural language processing, to me, is a really valuable tool. And that's where my lab is heading. In terms of aggregation techniques, physiology-based stuff is really cool. It's intuitive, and I like it. But it's not sufficient. Data-driven techniques like text mining and hierarchical clustering, I think, will be really important to help us know what can aggregate together and what can't, especially in cases where it's not very clear. All right, analytical techniques, we'll always need traditional statistics and mechanistic modeling, always. But I think two of the ones that are kind of the cutting edge are the de novo systems modeling, so things like dynamic meta-analysis, and then the machine learning and optimization, all right? But not any one of these is going to work on its own. It's going to take all of them together, all right? So regardless of how much I taught you about ALS, which I hope you did learn something, this is my main takeaway, is that when you think about neuropathology, many of you come from an engineering background, think about it from a systems perspective, all right? Many of these diseases are multifactorial, all right? And so even if we know exactly which gene causes that disease, like Charcot-Marie Tooth, well, why did that person get that gene? What caused that gene to do that, right? So, you know, coming at it from a systems perspective, and understanding that we each as scientists are one patch on a quilt, and we all have to be able to understand each other's stuff to get this comprehensive view, all right? So we need to shift from this conventional perspective that disease is caused by one factor, and if I can solve that one thing, then I cure the disease. That's just not how most diseases work. We need to have a systems level approach and perspective, all right? So these are some of the things that my lab is interested in doing other types of disease, and also different types of tools and applications. If you're interested in collaborating, I'm always open to that. Have to thank the funding, of course, NIH, NSF, and Georgia Tech. And finally, thank you for your attention, and I'll take a few questions. Thank you.
And really interesting. I've seen you talk before, but a lot of new stuff. So you mentioned that some of the analyses that you do, we could do to our own data. But it seems like you have a ton of data. I mean, you're aggregating so much data. How much data do you think you need to be able to perform some of the interesting analyses?
Well, that's dictated by the statistics of like your standard deviation or your interquartile range, depending on like whether it's normal or not normal, right? So that's how we determine, for example, how many samples do we need in order to have a certain like reproducibility on a relationship. So if you have a lot of heterogeneity in your data or a lot of variance, then obviously you need more, but I'm not telling you anything that you don't know. So kind of the standard rule of thumb that I use for these big types of models is, you know, if you're more than 2 standard deviations out, then you need to keep working. More sample size, change the way you're aggregating or something. So that's just, and that's just based on my experience, 2 standard deviations. So that's not like written in a book anywhere. So does that help? Other questions? No questions?
I have a question about Stephen Hawking.
Okay.
People,
So ALS is a diagnosis of exclusion, which means there's no litmus test that says, yes, I have ALS and no, I don't. We have to look at MRI, blood, muscular, action potential, what do you call it, EMGs. This is your line of work, right? EMGs, right? So they take all that and they try to determine. But the problem is there's so much overlap between ALS and primary lateral sclerosis and Pick's disease and all these kind of what we call a continuum of neuropathological diseases that there's no way to know for sure that Stephen Hawking has exactly ALS. Many people will argue that he doesn't because he's lived too long. But the also thing you have to understand is ALS on its own isn't fatal. It's fatal because they quit breathing or they get an infection. And Stephen Hawking has had access to amazing care. He's a smart man and he knows how to take care of himself. So, you know, people can be locked in their body. That doesn't mean they can't live longer, right? So I think it's kind of a combination that he's had access to good care and he's smart, and so he knows how to take care of himself, and he has a will to live. That's huge, right? So I can't say that he has ALS. He has something on the ALS spectrum, but whether it's the classical ALS or not, at the end of the day, I don't think it actually matters because I think when you come at it from the type of perspective that I'm advocating, then, you know, therapeutic-wise, we would approach it the same.
So you don't, I'm wondering if this approach would help actually distinguish a lot of the subtypes of various diseases that are difficult to distinguish. Would there be some test that you could throw a patient into your model and then say, okay, this particular person, because of whatever lab scores we got, suggests they should go down one line of treatment?
And that's the kind of the personalized medicine approach. I actually do have some heterogeneity models. I didn't go into detail about them, about ALS patients to kind of help predict, like when you're making your clinical trial for ALS, because that's a big problem with clinical trials, right? So we do have some models that are looking into that can take both the past, the epidemiology, the clinical metrics, and try to work with that. But they're mostly, they're data driven, right? So yes, I can basically make classes of patients that I think are similar, but I can't tell you mechanistically exactly why they're going to have, if that makes sense. But we can use this systems approach to be able to classify patients into a class, and we have been successful with that.
Is there a question down there?
Yes. Given that you're looking at this at a system level, homeostasis, and perturbations against that, are there any data visualization tools that you found particularly helpful in viewing it that way?
You mean like off-the-shelf tools or like?
Anything.
Okay.
Whatever you want to answer.
Well, I think when I take anybody's data set that I personally take and start to analyze, the first two things that I will run is a cross-correlation analysis and a principal component analysis, straight up. I think that's like gives you a lot of insights into local relationships and also the variance of your data right off the bat, right? So those are the first two things that I always do, and then from there, based upon whether it's normal, non-normal, whatever attributes of the data, then I go into more specific. But those are my favorite kind of cursory analysis to begin with.
Like you would model like a power plant or something like that, because you are looking at a systemic and doing, all right. I think I understand what you said.
Okay, well, I'll be glad to talk to you about it more afterwards.
So there's growing concern in the scientific community about lack of reproducibility in data and so on. And so one nice thing about what you're doing here is you're aggregating over lots and lots of different studies. So in some sense, aberrant studies it all sort of comes out in the wash, right? If there's data there that's less reliable, more reliable, you have so much of it that hopefully it
That's the hope.
It accounts for that, right? So I'm just wondering if you could think about this also, that sort of a post hoc analysis would be then to point back at particular studies that may have been less reliable or less reproducible than others. Like, could it be used as a diagnostic process?
It can. Like the example I gave was like with the strain, right? Before everybody was separating by gender instead of by strain, and it turns out that strain was more important, right? So you can use it as a diagnostic, but The trick is you have to have enough of a sample size to begin with to be able to differentiate something, right? So then whether it's good or bad is not, it's kind of subjective. Because sometimes, for example, data that maybe to an experimentalist would look dirty, I can actually use better because they gave me a standard deviation, a control, and a treated group. Versus another study that maybe was in a better journal and was from a, quote, better lab, maybe more respected lab. They didn't give me the standard deviation, and therefore I can do less with the data, right? So sometimes it's a matter of what you get and not just like the quality of the data itself.
How do you get published without a standard deviation?
Well, let me tell you that out of the 3,880 papers that I looked at for one of the last models that we did, about 22% did not have a control. Yep. Including papers in high-impact journals.
Yeah, so with how many hours it takes to aggregate all this data, and you said doing this automation, we could do natural language processing, but do you think it could be better to change how we actually publish the data?
I do think that that's the way of the future. Kind of like now, clinical trials are required to be archived and structured. Everybody has to structure their data and they have to archive them. It's required. Well, if we did something similar for scientific data, where if I'm going to publish open access, I have to upload my spreadsheet, and my spreadsheet has to be in this form, then it kind of would undermine all the need to do all this, right? But that's something that I think has to be, one, scientists have to see the need, and two, like journals would have to enforce, right? And we'll see. I mean, it used to be like open access was a pipe dream, and now everybody's going that way. So I'm hoping that with more awareness of what can be done, and also the fact, the other thing I've talked about other informaticists is I think there needs to be something where people can be cited based not only on their article, but their data. So say you upload your data to my database that I've created, and somebody wants to come use your data, then they can cite your data from my database, and it's like you get credit for it. Does that make sense? Because a lot of people don't want to share their data because they're afraid that they're going to like not get credit, right? And so I think there needs to be more ways to share data that somehow gives people credit, and then I think they would have like an incentive other than the greater good, which should be an incentive enough, but the greater good, you know, to upload their data in that form. Because a lot of the things that we do in our lab, had it been done up front, would not take nearly as long. It's just because we're trying to do it post hoc on a lot of things that are different.
So but you should be the one to tell us what form that should be in. We recently put some data on dryad.org because our open access journal
Required it.
And it is citable, but they didn't tell us how to make the data, like what format, so we just like threw it up there. So someone like you could definitely be involved in kind of telling us what do you need to make that data useful to you, and I'm wondering if you've worked with like PhysioNet, which is another NIH-supported database for people to put their data.
And those tools are helpful, but I think what happens is sometimes like it gets to almost too field specific, and so people want to get hung up on one aspect or another. And the thing is, when you're doing these type of really system level models, you know, that detail is awesome, but at some point you have to aggregate. And so, like, you know, for example, the way we're doing our database now, it's not ALS specific. It can be done with any mouse model, okay? They all measure the same way. It's a transgenic mouse, you do this, that, and the other. Some things are different, maybe like rotarod versus Morse water maze, right? But you plot it the same way, right? So it's being able to, I think, teach people that it's okay to compact your data to a form that maybe seemed over aggregated to you, but works better like for the greater good of doing complex models.
Last question. So just to follow up on that comment, NINDS and their Common Data Elements project are implementing preclinical data elements. So as you know, there's a lot of clinical.
And that's what we used for our clinical ALS database was the common elements. Yeah.
Right. So there's several preclinical ones that are getting ready to launch that would deliver standard format and data elements for people. So that's modeled after the clinical data. Now, one of the things I will comment about it is it's a database, right? So there hasn't been a whole lot of thought into the
Right, because they think that that's the responsibility of who wants to use it. Right, which is, but at the same time, there has to be a kind of halfway thing, right? Where it shouldn't be the responsibility of just the person dumping the data to make it perfect for the person analyzing it. Otherwise, the person dumping it could analyze it, because a lot of the stuff I do, I can teach you how to do pretty fast, right? I mean, it's just a lot of standard tools used in a certain way. So there does have to be a marriage of that. But I think a lot of times, like, people who figure out how to aggregate for common elements or whatnot, I know, like, for the clinical one, they didn't ask any informaticists in the room to be there. Like, I talked about this with the ALS people. It was all clinicians, and they didn't know what needed to be needed. So I think it's like kind of that understanding that you have to have different viewpoints there, so that when we make these decisions, we can collectively kind of give and take, and hopefully do better than what Congress does. So.
All right, Vincent, thank you.
Thank you.